Okay, students, so I'll be explaining about the different experiments that you have to write in your record book. So we just started with the first experiment that I told you. The first experiment you have to write is the experiment of the stomatal distribution. So you have to write experiment number 1. Then directly you can write, instead of object I told you, you can write the aim and write the sentence. After aim you come to the requirements. Okay. Then after requirements, you can directly move to the next part, which is the procedure. Okay, so write the procedure as it is given. There are 7 points total given for the procedure. After procedure, you can write the observation and precautions. Okay, then this first diagram only you need to draw in the left side of the first experiment. Okay, so that is about the first experiment you have to write. Now the second experiment, you will write this paper chromatography. So you have to write the heading experiment number 2, then write the aim here as given in the sentence, then write the requirements, then you have to write the procedure. Okay, write the procedure and then observation, calculation, that is all on the right side of the record. And then you can draw this column. I told you, if you want you can draw it on the left side or right side, that depends on you. If you are using the left side, you have to draw with pencil only. If you are drawing in right side, you can draw with pen. Then write the result and any two precautions. Any two precautions you have to select and write. Okay, so that is the writing part on the right side, and only need to draw this diagram. This diagram only you have to draw on the left side of the experiment. Okay, so that is your second experiment, that is the paper chromatography. Okay, now your third experiment is to test the presence of sugar in urine. This is your third experiment, that is coming on page number 79. You have to write experiment number 3. Aim is to detect the presence of sugar or glucose in urine. Write the requirements, then write the procedure and observation, and write the conclusion. Okay, and you need to draw only this particular figure. Draw only this particular figure on the left side, and precautions, it is written here same as for the experiment 10.1. So please go back to the experiment 10.1 precaution. Here they have written the two precautions. That same precautions only you will write for this glucose test precaution. Okay, you need not write this experiment, only you are taking the two precautions from this experiment. We are writing only this sugar in urine experiment. Okay, so from the precaution, for writing the precaution, you have to take it from this experiment as they have written it. It is given in 10.1. Okay, and draw only the single diagram. Okay, so in the previous class, I had told you are also doing that next experiment, that is test for the presence of albumin in urine, but we are not writing it in the record because we don't have this reagents in the lab. Okay, so we are not doing that, and so we are not writing that also. So as this experiment number 3, you will have only one experiment to be written, that is the test to detect the presence of sugar in urine. Need not write this albumin test, that is not there. So please put an into mark, that is not needed. Okay. Okay, so with these 3 experiments, you are finishing off your core experiments. Okay, so you have to take another new page on the right side and write there experiments for spotting, or simply you can give the heading spotting. Okay, after writing spotting, take the next new page and you have to write the experiment number 1. Again, you are starting from 1 onwards in the spotting heading. Okay, so I told you in earlier class, I told you to write parts of a compound microscope, but you need not write that in your record, but you can study this different parts of the compound microscope for your understanding of the microscope. You need not write the same in the record book. So you can just read and understand the parts of the microscope because it is needed for you to handle the microscope. So you need not copy this to the record book. So instead of that, your first experiment coming in the spotters will be to study the plant specimen with reasons. That is, the first experiment is bacteria which is coming in page number 89 here. Okay, so you will give the heading. So spotters, your first experiment number 1 is aim. You can write the aim here, to study the different specimens and identification with reasons. This can be written as the aim of the experiment, because we are not writing all that is given as aim here. It is given so many names. We are only selecting and writing. So you need not write this as the aim. Better you can write this as the aim of the experiment number 1 in your spotters or spotting. Okay, so this will be your aim. Then you can write the requirements as given. Then okay, you can write the procedure. You can write the procedure, and then I told you we are only selecting some organisms. So first organism which I have selected is the bacteria. Okay, so you have to write heading. After writing the procedure, you have to write number 1 bacteria. You can write the classification, and you need to write any 2 comments. Okay, you have to write only any 2 comments. You can select any 2 comments and write only 2 comments, and you have to draw this picture on the left side, only this picture. Have you understood? So write number 1 bacteria, write the classification, write any 2 comments on bacteria, and draw this diagram on the left side of the writing of bacteria. Then next organism we have selected is Spirogyra. You are writing only number 2, you have writing Spirogyra. Write the classification, write any 2 comments, only 2 comments are required, and you can draw the second diagram. Okay, which is showing all the markings. Write any 2 comments and draw this particular diagram. Okay, so that first one is bacteria and second one is Spirogyra. Then the next third organism we have selected is mushroom. Third organism is mushroom. So you have to write the classification, write any 2 comments, and draw this first diagram. Okay, write the classification, 2 comments, and draw this diagram on the left side. After mushroom, we are coming on to dicot plant and monocot plant. Okay, so the next one we are writing is this one, dicot plant, or you can write first monocot and then dicot as you like. So dicot plant, if you are writing, you have to write number. What is your number? It will be 1 2 3 4 for this dicot plant. If you write number 4, then write the name, that is mustard plant, write the classification, and write any 2 comments and draw this diagram that is showing the all parts of the dicot plant. This diagram alone you have to draw, and write only 2 comments. Write only 2 comments, nothing else. Diagnostic features, nothing you need to write, only 2 comments. Okay, then take the next one, that is the monocot plant. In this monocot plant, they have given some plant which is not familiar to us. It's okay, you can copy the same thing, but here they have made one mistake. Instead of subclass monocotyledone, they have written here dicotyledone. So please correct it and write monocotyledone. Okay, then you can copy the same thing, write any 2 comments only, no diagnostic features required, and you can draw this particular figure. Okay, this figure for the monocot plant. So with that, our the plant specimens are over. And last one you have to write is lichen. With lichen you are ending that plant specimen features. So write the number accordingly, write lichen, and then write any 2 comments on lichens, and then you can draw any one picture. This picture you can draw. Okay, this picture you can draw, and you need not mark anything over there. Just draw this picture and just write under that is foliose lichen. No need to mark anything, because we are not studying in detail about these things. So just draw this picture, write under that foliose lichen, and write any 2 comments, and with that you will finish your plant specimens. Okay, the next is your animal specimen. So once you write spotting experiment number 1, identification and classification of as plant specimen, then 1 2 3 4 5 like that continuously you are writing. You need not start everything in a new page. Continuously you can write till you reach your lichen. It goes continuously. No need to start new pages for each heading. Okay, so you can write continuously till you reach lichen. It is over. Now you have to take a new page for starting the animal specimen. Okay, so you're writing the heading experiment number 2, and this can be written as the aim of the experiment: study of characters of animal specimens and identification with reasons. This will be your aim of the experiment. Okay, so you're writing experiment number 2 and writing this as the aim. Then you can write the requirements, you can write the procedure. Okay, after writing the procedure, like you have written for the plant specimen, you will write number 1. So here we are not writing amoeba, we are moving on to hydra. The first one we are writing is hydra. So you can take hydra as number 1, write the classification, write and select any 2 comments you have to select, draw this diagram, no need of any diagnostic features. Okay, just select any 2 comments, write the heading hydra, classification, and write 2 comments and this diagram on the left side. After hydra, the next is liver fluke. This you have to write. Second organism is liver fluke, classification, any 2 comments, and you can either draw small one or big one, that is according to you. Okay, small one is the yes, that is 2 species are given here. You can draw any one according to your wish, no problem. And then you have to label the parts. If you want to draw this or draw this, that's according to your wish. Only one diagram you need to draw it. Okay, so that is about liver fluke. Then our next organism is Ascaris. Ascaris, that roundworm, write the classification, write 2 comments, and you have to draw 2 diagrams. You draw because this is showing the male Ascaris, this is the female. Both specimens we have in the lab, so you can better draw the 2 diagrams. Just draw these 2 diagrams, no need to draw the small one, this male one and the female one, and label the parts. That's all, and select any 2 comments. After that, the next organism is leech. Leech also write the heading, classification, write any 2 comments only, write any 2 comments only, and then you can draw the diagram. You can draw this diagram. I think this will be simple. This diagram you have to draw and label the parts. Okay, after the leech you have to draw the silkworm, that is yes, this one. From the arthropoda we are drawing the silkworm. Write the classification, select any 2 comments, and you can draw the life cycle of this silkworm. Okay, on the left side of the page. After silkworm, the next is that rohu fish. This is the rohu fish heading. Write the heading and write the classification. Take any 2 comments, then draw the diagram of the fish. Okay, next one is frog. Write the heading, classification, draw the diagram, write any 2 comments. Next one is lizard. Write the heading, classification, 2 comments, and the diagram. Then after that you have to write. Okay, so you can either write this wall lizard. Okay, this is the chameleon, so you need not write this chameleon, garden lizard, we don't want that. We will write this one. Okay, this is our normal wall lizard, so you will write Hemidactylus, that lizard, that write the classification, comments, and the picture. You need not write this one, this is that chameleon. Okay, that is called the garden lizard, so need not write this one. Instead of that we are writing only this lizard. Then pigeon, write the classification, write the scientific name, classification, 2 comments, and draw the figure. And last is in the mammalian, you have to write the rabbit, classification, 2 comments, and draw the diagram. Okay, so all this will go continuously. All the animal specimens you can write continuously. No need to have fresh pages for each heading. Okay, but wherever that particular heading and writing is coming, just the left side of that you have to draw the picture. Okay, so that is all about your animal specimens. Now the next one is the mitosis. Okay students, so the last experiment you are going to write is the mitosis in onion root tip cells. No need to write in animal cells, you just give the heading study of mitosis in onion root tip cells. Don't write animal cell, we are writing only in the plant cell. So write only in the onion root tip. Okay, so you can write this as the aim itself, to study the various stages of mitosis in the onion root tip from the permanent slides. You can write the requirements, procedure, observations, and you have no need to draw this diagram. No need to draw this diagram. Write all as it is given. Then you have to draw these diagrams on the left side. Okay, draw these diagrams only on the left side. Then continue with precautions. Till the precautions you have to write on the right side. Okay, so all these diagrams will come in the left side. These writings will come on the right side. No need to draw this diagram. Is that clear to you? So write the heading study of mitosis in onion root tip cells. No need to add the animal cells because we are writing only in the plant cells, not in the animal cells. So the last experiment is this one. With that you will end your practicals. Okay, so this much you have to write in the record book. So you have to buy the record book from outside as it is not available in the school. So when you're getting time, go and purchase the record book and start writing at least one experiment. If you start writing daily, then it won't be a much burden to you. Okay, so I'll be asking you by December. By the end of December only I'll be asking you to show me the record books. Okay, so by that time you have to complete it. So record is a must for 11th standard, otherwise we would have skipped it. So we don't know whether we will be able to do it or not, we don't know, but as far as possible I'll be showing these experiments through virtual labs. Okay, so you will get an idea of how to do the experiments. So record writing, you have to keep it aside. As I told you, you can use a blue pen for writing and black pens for underlining, and on the left side always use sharpened pencils only for writing the headings of the diagram, for marking the diagram, everything. Okay, so I think 200 pages record will be needed, I think, because these many experiments you have to write. So you can purchase one record. I think that classmates record will be enough. Okay, so start writing as you are getting your time, and if you have any doubt you can ask when we have our next class, or in between if you want you can WhatsApp or call me and clear your doubts. Those who have the record can start writing. Okay, I hope it is clear to you. Thank you.